So now we will talk about the mRNA and the tRNA. The mRNA, or the messenger RNA, consists of thousands of mononucleotides. It is a long strand arranged in a single helix. Its sequence is determined by the sequence of DNA that is transcribed to make this RNA. The process that does this is called transcription, which we will discuss next. Once formed, the mRNA leaves the nucleus through the nuclear pores and enters to the cytoplasm and associates with ribosomes. The mRNA at the ribosomes is used as a template for protein synthesis. The structure of mRNA serves its function because it has information in the form of codons that are complementary to the DNA codons. As for transfer RNA, it is a small molecule made up of around 80 nucleotides. It is also single-stranded. However, it is folded into a clover leaf shape. One end will extend beyond the other. The tRNA attaches to an amino acid, and we have many types of tRNA. Every type is specific to one amino acid. At the opposite end of the tRNA molecule, the opposite end to where the amino acid is bound, we have three bases known as the anticodon. These are going to be complementary to codons. We have as many types of tRNA as we have coding triplets, since the genetic code is degenerate. So let's say one amino acid will be coded by 3 DNA codons, then 3 kinds of tRNA will be bound to this amino acid. In RNA, we don't have thymine, we have uracil instead. So the base pairings in RNA are guanine binds with cytosine, like in DNA. However, adenine in RNA will bind with uracil in RNA. So when we have protein synthesis, which is the process of translation of mRNA into polypeptides, the anticodon pairs with the codon on the mRNA, bringing with it the amino acid attached to the other end. And this is how we start synthesizing the amino acids into a polypeptide chain.